Last part of chapter 10. Now we're actually going to make the protein. So just to remind you what we've been going on, we have the DNA in the nucleus. It gets unwound and copied by RNA polymerase. That's called transcription. Then we have pre-RNA, mRNA, that gets trimmed down. We take out the introns, we splice the exons together, add the 5' G cap poly A tail, gets signaled to leave the nucleus through the nuclear pores, and now we have translation. We're going to have the ribosome find that mRNA, and we are going to turn it into a protein. So here's a X-ray crystal structure protein computer model of ribosome. Hard to see the two different subunits, but there's a small part up here, the small subunit, and then there's a second part down here called the large subunit. This is a bacterial ribosome because they're showing you an antibiotic tetracycline that can bind in there and block protein synthesis in bacteria. Eukaryotes also have ribosomes that look very similar to this. They're just a slightly different size, so we don't get interfered with if we take tetracycline in our eukaryotic cells. It only hits the bacterial ribosomes. I put this picture here mostly not to talk about the bacterial ribosome, but to just point out how the structure of the ribosome in both eukaryotes and prokaryotes is made up of protein and RNA. So ribosomal RNA, or rRNA, is coded for in the DNA, gets turned into RNA in the nucleolus area of the nucleus, and then that RNA finds protein and makes ribosomes. The small subunit and the large subunit are assembled separately in the nucleus, and then they have to fit through a nuclear pore, just like the other stuff does, get out and exported to the cytoplasm. So that's sort of like a background thing that's always going on in the cell. They show you here that the RNA portions of the ribosome are in green, so they're this more stringy stuff. And then we start to recognize some protein structure, a lot of alpha helix going on, maybe a little beta pleated sheet there, but mostly alpha helices you see here of the protein that's bound to the ribosomal RNA. In any event, that's what the assembled RNA, our ribosome would look like. So now here's what's happening. We're going to have the mRNA arrive in the cytoplasm, and then it finds, actually the small subunit first, finds an AUG, and then that attracts the large subunit. And then when these two pieces come together, and here they have more of a space-filling model, it creates 3 locations in the center where the action takes place. The E part is called the E part because it's for exit. It's the last place. The P part is where the peptide will be growing, so this is sort of the intermediate site. And the A site you could call the acceptor site. So this is where the newest tRNA will be coming in to bind. And you're going to see that as we go. So these are the areas that are being the mRNA is being fed through, and the action will be going on there. Seems like the same picture, so sorry about that. Here we go. So what happens? Again, the mRNA gets out into the cytoplasm. The small subunit scans that mRNA until it finds what's called the appropriate context of an AUG. They've scanned a lot of mRNA sequences, and that AUG is that first methionine, it's that first triplet codon, and then it tends to have A's and G's at a particular spacing 5' to that AUG that makes it in the correct context, and the ribosome knows that. If you picked up an AUG, for example, that's out of frame, imagine you would have junk protein translated later on. So there's a lot of information coded in that mRNA to make this a likely place for the small subunit to bind. And that small subunit binds that correct AUG, the first tRNA comes in. So here's the tRNA upside down from the pictures we've been looking at. Methionine is attached to that tRNA because that's always the first amino acid put in there. Here's AUG in the mRNA, so the anticodon is going to read, if we're looking at it in this direction, UAC. But remember, this is going to be 3' and that's going to be 5'. They still have the anti-parallel binding thing going on. Don't get too caught up in that, though. Anyway, that binds. This signals the large subunit to come, and it comes and it attaches in a way so that this current methionine is going to be in this P station. So that's initiation. the next step is going to be elongation, where we basically repeat bringing in a new tRNA that matches that codon. Here we got that anticodon matching it. That's going to come in the A site. Then the ribosome essentially is not just structural, it's not just a platform to be building on, but it's also an enzyme. So the enzyme feature of this is going to transfer the methionine that's here to this proline and make our covalent peptide bond. So that enzyme complex in the ribosome does this. This costs energy. This costs 1 GTP. So when we put every amino acid in, it costs energy to do this. Translate that. Then the whole ribosome kind of shifts over so that your new guy is now bound in the P site. It's P site because it's the growing peptide chain. Now we have 2 amino acids hooked up there. And then we can keep doing that. You can see we have the next one coming in now. This one matches up and it's a tryptophan. I'm sorry, a tyrosine, a TYR. It's going to bind that. Again, we're going to do that same reaction. Move this guy over to this one and then grow that long chain. So it's just going to keep chugging along. And they think it's actually the ribosome that chugs along and moves along that RNA molecule. Then you hit that magic stop codon. So in this case, I think they're showing it looks like a UAA. So the thing that recognizes the stop codon is something that's not a tRNA now. It's a release factor. So it's a protein that binds and signals the end of the protein synthesis. That catalyzes a reaction to release that polypeptide chain or protein that's been growing. Everything falls apart, and voila, you start over again. And there are ribosomes floating around as different subunits looking to just diffuse and bump into mRNA. So there is no real planned way of this happening. It's just a pure diffusion thing. So just thinking about, there are signals that start and stop transcription and translation. So transcription, remember, that's turning the DNA into RNA. You started out by binding the promoter. Remember, RNA polymerase likes to find a promoter. And then there is a termination signal in the DNA that tells you to shut down. And when you're translating it, when you're going from RNA to protein, we have that AUG start codon always that's coded in the mRNA, brings in a methionine. And then there are the 3 special stop codons that tell you to stop. So at any given time that the RNA is out there, it could be having several ribosomes attached to it. So they've got pictures of this. So that's your evidence there of an mRNA in the middle and these little ribosomes. And you see as you go down the line, they've got longer proteins hanging off them. So they can find these in the cytoplasm and they know that that's what's going on. mRNAs have a lifetime. We'll talk about that as we in the next chapter. We talk about regulating how many proteins are being made, both at the DNA level and at the RNA level and at the protein level. All different ways to control how many proteins you're going to actually kick off of one message. And then the last idea to talk about in this chapter is really actually one of the coolest ideas, I think, in cell biology. And that's that proteins have embedded in them sometimes a signal to go to different organelles. We danced around this. We talked about the parts of the cell a long time ago. You know that if we go on this picture over here, if the protein is destined to leave the cell by exocytosis, so maybe it's a hormone, like insulin actually, that's supposed to leave the cell, it has to transit through the rough ER, and then the Golgi, and bleb off on our vesicles, blah, 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 and then fuse with the membrane. So how does a protein know it's supposed to get there? Well, there's a signal sequence in the initial part of when you're making the protein that actually drags it to a docking station. I don't have a picture of that, I think, coming up. That helps that protein get secreted into the lumen of the rough ER. Remember, the rough ER is rough because you have the ribosomes dotting on it. And it's actually this piece of information right here that just is beyond the distance, physical space of the ribosome that now can stick out and attract to a docking station here. And then it goes through its whole thing. You similarly need to have proteins that are going to get imported into the nucleus, into peroxisomes, into mitochondria, and into the chloroplasts, because they have to do their job in there. There's a nuclear targeting signal, and I believe that the book explains an example of an experiment that was done where they move that signal from one protein to another and prove that it is necessary and sufficient to target a protein into the nucleus. There were some really fascinating experiments done with that. It's similarly, there's a targeting signal that brings you into the mitochondria. Remember, the mitochondria has 2 membranes, so it's actually a very complex signal that codes to get the protein in there. The gene that I cloned when I was in grad school ages ago, we wanted it to be in another compartment in the cell. And when I cloned it, I proved, unfortunately, that it had the mitochondria targeting signal on it. So that's why it's a little bit near and dear to my heart. Still had never been cloned before, and people were really looking for that protein and how it could get into the mitochondria, and I found that one. It just wasn't doing the job that we thought it was going to do. But anyway, signals send things to the different parts of the cell. Again, I always like to note the Nobel Prizes. Nobel Prize was won by Günter Blobel. He's a German-born. I believe he still does his work in the United States now at the Rockefeller Institute, maybe. He got a Nobel Prize in about 98, 99 for deciphering a lot of these codes and how proteins get targeted to different parts of the cell. So again, I promised you a better picture of the ER. So here's our protein getting made on the ribosome. Dark green is the mRNA, little balls are the protein. So it turns out that when you get to about 12 to 18 amino acids long, it's just enough for that to go and bleb out past the edge of the ribosome. And that's how long it has to be. Now this signal gets recognized by a docking complex. This is an incredibly complicated group of proteins. It's not just two little blobs. It's a very complicated little gate. It binds that, and then it drags it into the lumen of the ER. And sometimes, if this would be a transmembrane protein, it would get stuck there. This one looks like it's going to be a secreted protein, so it's going to go into the lumen and then go on its merry way like we know. This was the experiment, again, of the nuclear localization signal. I'm not going to talk through it because you can read that in the book. And then lastly, sometimes after proteins are made, they get chopped up into pieces. One example of that is insulin. Insulin gets made, it gets put into the ER, it gets sugars attached to it, glycosylation, sugars attached to it, and insulin gets chopped into pieces. It's called pro-insulin when it's first made, then it's called pro-insulin when you chop off one piece, and then it's called insulin when you chop off the last piece. So things still happen to proteins after they're finished, and that's called post-translational processing. The little red things are little sugar branches. We know that those start to get added in the rough ER, and then they get trimmed and modified in the Golgi. The sugars are going to be important with different proteins that do different jobs. A lot of times it's for cell identification or some binding sites or maybe some solubility issues, but there you can put sugars on proteins. And then lastly, you remember phosphates through cell signaling. We can activate and deactivate proteins by the addition or the removal of phosphates, and that's something that can happen after a protein's made. That's the end of chapter 10. Moving on pretty quickly, we'll get to chapter 11 soon, and that's going to be all about how we control at the DNA level and then later how long proteins are around and if they ever get made.